Hello friends, in this video we'll be talking about the replication termination or the termination or ending of the DNA replication process in bacteria like E. coli. Now there are several different mechanisms we have talked about the replication termination, but in this case we will be talking about a very interesting and new concept about the replication and termination. Now that this termination is done with the help of a protein called TAS protein and tar protein, and there is a site for binding of this protein. No, site is tar and the protein is called the TAS protein. Okay, anyways, now let me talk about it. Let me take a color. Okay, fine. Now you can see in this picture very carefully that the termination of this replication is important because we need our DNA sequence to be synthesized properly. When it is copied completely, then it must leave that sequence. Now how can this be at the end? If you look at this case, we can find this helicase or DNA helicase is the enzyme which is unwinding these two strands of the DNA, and DNA helicase is going through this portion which is called the replication fork. So this is the replication fork, and DNA helicase is clearing the passage for polymerase to sit on and go on replication process. But right after that, this DNA helicase can be blocked only by one type of protein DNA interaction, and that is the interaction between TAS and TAR protein. So that's why the complex of TAS TAR protein is really important before stopping the DNA replication cycle. Now in this case what we are looking at, the helicase is going on from this 5' site at the below, and this is a TAS protein which is binding to the TAR region of the DNA. TAR region means the termination region at the site of the DNA. So there are particular regions in the circular DNA of E. coli or a bacterial cell. There is one point which is called ODC or origin of replication, and there are also totally almost 180-degree opposite direction. There is another site which is called the termination site. Now both of the sites are important because from this origin site DNA replication begins, and at this termination site DNA replication ends. Now in this termination site a protein called TAS will bind, and binding of this protein TAS to this termination site can lead up to the blockage of this DNA path, blockage of the path of DNA helicase movement. Now DNA helicase move on from this 5' end towards this direction, but when this protein TAS come in and bind to the TAR sequence, it bind in so much tightly that sometimes this helicase cannot cleave it away from this cell. Now in this picture what we are looking at, the replication fork is moving on to this direction. So let me take another color. Anyways, it is moving on to this direction, but right after the binding of TAS protein on this termination region, this it blocks the path for this helicase, and helicase can go on to this place, and after going on to as helicase move on to this direction much closer to this TAS-TAR complex, it creates the pressure onto this TAS-TAR complex, and it eventually leads to the separation of TAS protein from the termination region, and this protein falls off, and as a result fork will move on to this direction. So no termination happen in this case. So this end or the end of 5' at the below is called the permissive end for DNA helicase to move on, and as helicase is moving on in this direction, we can say that polymerase will also move on to this direction. Okay, so that's understood. Now in the second scenario, what can happen if this TAS-TAR protein binds to the same site, but the helicase is coming from the top region, from this 5' end, from the top region of the 5' end. Right after coming to this site, it must go on to this direction to dissociate TAS protein from this TAR region. But what happened when this helicase attack from the top of 5' end site? In those cases, TAS protein can melt the base onto this site, and this is a cytosine base which is being melting means the denaturation of this DNA double strand, and not only that but also the base is flipped towards the outside of this DNA strand. Okay, now this base is called C6, that means the cytosine 6. This is not the carbon number, this is the cytosine base number 6 at this position. So if we think about the positioning from 0 and then +1 +2 +3, so this is the +6 position of nucleotide, and this nucleotide is cytosine. That's why it's called the cytosine at 6 or C6. Now this C6 position is melted and flipped onto the opposite side of the strand, and as a result of this base flipping, it actually blocks, or it actually attacks this TAS protein, or block the TAS protein onto this termination site, and it makes the binding of TAS protein towards the termination site much more stronger, that this helicase cannot rip that one apart from this place. Now in previous case we can see the movement of helicase creates a strain onto this DNA strand. As a result, TAS protein leave the termination site and DNA replication will go on. But in this case, when this when this helicase is coming from the non-permissive end, or the top side of the 5' end, it melts a region of this TAS protein, of this TAS interaction of this DNA sequence, which is C6 region, and this base is flipped outside. As a result of this base flipping, this helicase cannot move on, as well as this TAS protein bind to the termination site pretty much more stronger in affinity than the previous time. As a result, this helicase is not able to dissociate this one out, and as a result it remains in this remains in the binding condition with this DNA. Okay, so this is called the non-permissive end, or the non-permissive end for this DNA helicase, because helicase got two different ends. One is permissive, one is non-permissive. Now it is called permissive because it permits the helicase to move on, but this end does not permit the helicase to move on. That's why it's called the non-permissive end. Okay, so this is how the replication is terminated utilizing TAS and TAR proteins. It's completely new concept, like it has been published in 2006 paper, and this is also called a molecular mousetrap for termination of the replication. Okay.